So I'm going to take you on a little tour today through research that is ongoing in my lab here at Thayer, and focusing on the Hello? Now we've got it. Okay. So I'm going to take you on a little tour of research that I'm doing in my lab here at Thayer. And I call the lab the Multimodal Neuroimaging Lab. Now, there are three main areas of thrust in this lab. One is on instrumentation development, the other on algorithm development, and translational research. So we're going to use this as a little roadmap to guide you through what I've been up to. So the research that I'm doing to develop brain imaging technologies has a context, just as all of the research does here. So we'll start there and ask why study brain function. What motivates me most of all is the need to gain a deeper insight into all of these different brain disorders, such as Alzheimer's disease, Parkinson's disease, schizophrenia, bipolar illnesses, autism, epilepsy, attention deficit hyperactivity, traumatic brain injury. So there's a whole long list and many more where understanding brain function is fundamental to advancing and improving the human condition. There's an additional reason, which is to get answers, to get closer to answering fundamentally profound questions about human cognition and human experience. So this is part of a very long-term endeavor by humanity, and I'm looking to do my part to move this along forward. So let's think about brain disorders. Brain disorders globally have a major impact. So including neurologic disorders, substance abuse, mental, behavioral disorders. Brain disorders are the number one source of disability globally. They account for 22% of the disability-adjusted life years from all causes between 15 and 49-year-olds. So that's a major issue globally. In fact, it's going to be the largest cost of chronic non-communicable diseases over the next 20 years. Here in the U.S., these are also critically important issues. If we just picked on one of these disorders, Alzheimer's disease, that's in 2013 had a direct cost in current U.S. dollars of $203 billion, and that's projected to rise up to $1.2 trillion by 2050. So this is just this one disorder has a major impact on our country. Now, this is, of course, fairly widely recognized. The NIH, with the White House and other private partnerships, has launched the BRAIN Initiative, which stands for Brain Research through Advancing Innovative Neurotechnologies. Now, when I heard about this announcement, I was thrilled, in fact, because I believe, as is outlined in the BRAIN Initiative, that the key to moving us forward into this next phase of new understanding about the brain hinges on developing the new technologies that will take us there. So here's a quote from Incel Landis and Francis Collins, who's the director currently of the NIH, that now is the time to create the next generation of tools by harnessing advances in diverse disciplines from engineering to computational science. And this is really a shift in focus from a focus on wet lab bio to the need to advance these engineering tools and computational tools to move forward. Really, we need all of them, and this is the focus of the BRAIN Initiative. So not only is studying the brain important globally and important in this country, it's also incredibly challenging. So I thought I would focus in on here the level of complexity that you find in a single fMRI voxel. So this is one little chunk of brain that's about 3 millimeters by 3 millimeters by 6 millimeters in size. So within that small region of brain, you've got 5.5 million neurons. You've got 50 billion synapses, 22 kilometers of dendrites. These are the connectors between the neurons, and 220 kilometers of axons. So this is a tremendous amount of circuitry compressed into that small bit of brain. But that's not even the whole picture. In addition to the complexity of neurons, they don't live in a vacuum. There's a whole environment, a whole microenvironment supporting those neurons and their behaviors. And so that we call the Neurovascular Unit. So in addition to the neurons we've got coming down here, we have synapses between those neurons, also with astrocytes. These are helper cells. We're not able to measure them very easily from outside of the brain or even down in locally, but they're very active in supporting the activity of the neurons. And we've got to get nutrients and blood, gases, oxygen to these neurons and take away those metabolic byproducts. And all of this is really intricately linked together so that there is a responsivity to the whole environment, to the activity of the brain. So it's not just the neurons that are active. The whole neurovascular unit is active dynamically during brain function. So what about the technologies? What are the ranges of technologies that are used to study the brain and measure brain function? So I thought I'd start with one of my favorites as I cruised around through history. This one goes to the 17th century. It's an engraving, The Physician Curing Fantasy. So here we've got a physician, I presume, pushing the subject into some sort of furnace, and all the fantasies are shooting up out of the top. So I thought this is, fortunately, we've moved a long way from this engraving. There's something else going on in the background here that I find even more terrifying. I don't really know what's going on. So modern technologies and methods are significantly improved beyond that engraving. We've got really an alphabet soup of different types of methods and modalities of imaging the brain. PET, positron emission tomography, computed tomography, diffusion tensor imaging is one of the variations of using the MRI machine to study tracts and projections through the brain. EEG, the electroencephalography, electrocorticography, when you bring your electrodes down on the brain, recordings made down with needle depth electrodes in the brain. There are whole ranges of these different techniques. Also optical techniques, magnetic imaging techniques, ways of imaging pharmacological effects in the brain. So there's this very, very wide range of technologies and methods that are used. Now, despite all of these existing technologies, there are gaps. There are major gaps that limit our ability to advance at the rate that we'd like to see to unlock these mysteries of the brain, understand what's going on in the diseases and disorders. So, for example, most technologies measure a single aspect of the brain physiology. It's going to measure a neural electrical signal, a metabolic signal, or so forth, but they're not showing us the relationships, often very complex relationships, between those physiological dynamics. Current technologies measure at typically a single spatial temporal scale. It's going to be either like that fMRI Voxel, a few millimeters in scale, or maybe larger for, say, an EEG technique and so forth, or maybe very, very small down at the cellular level with a needle electrode. But bridging between those scales, both in space and in time, remains one of the critical gaps. In addition, these techniques will measure fine details of the anatomy and the physiology, but relating those measurements to human experience, to human disease, that requires the insights of the experimenter and the methods that you're going to use to interpret those signals. And we're actually sitting already in a position of having a major gap, a gulf between what we can measure and what we can interpret and understand. So this is another one of the gaps. So now I'm going to take and generalize very broadly here and say you could take the majority of the brain imaging techniques and categorize them into methods that measure signals related to neurons, neural-related signals, and signals related to blood and blood dynamics in the brain. Here I've got an image of showing the outer surface of the cerebral cortex, and these are the larger blood vessels that you have going up in and around the brain. So broadly speaking, here are a couple of prime candidates for measuring neural signals. If we're talking about non-invasive measurement, the EEG, where you put one of these caps with electrodes on your head and measure those electrical potentials on the scalp. Another technique would be the MEG, or the magnetoencephalography technique, where you measure the magnetic fields coming from the brain. We'll discuss these a little bit more as I go forward, but these are both based on neuronal signals. Blood-related signals are being measured by the fMRI, where you have changes in blood oxygen affecting the local magnetic environment, and that drives the contrast you get with your fMRI machine. Or, for example, this near-infrared diffuse optical tomography technique you'll be hearing more about through my talk, which is sensitive to changes in the blood oxygen and blood volume in the brain. So when you're using these blood signals, you're then inferring neural activity based on that link between the neural activity and the hemodynamics. That link, that neurovascular coupling link, is what enables studies using an fMRI machine to say, oh, well, this is representative of brain function. And not understanding that connection creates something of a challenge in interpretation. So in my lab, what we focus on is really this first fundamental question of how do these signals relate? If we have some technologies that measure neural signals and others that measure blood signals, how do we relate those two technologies to one another? So this brings us to instrumentation development and so forth. I'm going to now walk you through some of the ways that we're using technology to bridge these gaps. So the first enabling technology I'm going to go into in a bit more detail is the combination of near-infrared spectroscopy and electroencephalography, or NIRS EEG. So what is NIRS? First, it does go by another name. When you're doing imaging, it's often called diffuse optical tomography, or DOT. So I may use those interchangeably. The idea here is that you have an array of pulse oximeters. Many of you have probably had a cuff put on your finger or on your ear to measure blood oxygen changes. Imagine a whole array of those put onto your head, and now you've got a near-infrared spectroscopy machine. So it's going to provide an image of the blood oxygen levels on the brain, and that's going to be animated over time. So you can observe the blood oxygen dynamics on the brain with this technique. You do it by exploiting what's called an optical window. So if we consider light in the near-infrared range, so say in the wavelengths of 700 nanometers to about 900 nanometers, those sort of wavelengths of light, they're able to travel more deeply through biological tissue. Really, they're scattering around through the tissue. And that's because the absorption by water is lower than for other colors of light. So if you pick a couple of wavelengths to use, for example, 690 and 830 nanometers or others in between, you can then resolve concentration changes in oxygenated and deoxygenated hemoglobin based on known properties of how they absorb light. So to get the light down into the head, we use things called optodes. This is showing some ray tracing of photons being reflected off of a prism and going through a small hemispherical lens before that light is deposited down into the head. So this is an axial slice of the head where we have, coronal slice of the head where we have light being deposited here at the red dot, and then you collect light that has scattered around down in the tissue some distance away, and that would be, say, at this blue dot location. And when the light has traveled down between those two, if you have a change in the amount of an absorber, like the hemoglobin, you can detect that concentration change. So here I am wearing a cap populated with these optodes, shining light in various locations on my head and measuring back the changes in the amount of light. And we use that to then infer the oxygenation in the brain. So here's the instrument. All the laser sources and photon counters are here in the machine, the computer and display, and off we go. Now this head probe that we've built in the lab has these locations for the optodes, but it also has positions for electrodes. So we can also measure with the EEG. And with the EEG, what you're doing is measuring potential differences, voltages between positions on the scalp, and from that you're inferring electrical activity down in the neurons. So we've taken these two and integrated them into a single probe that is easier to use, more repeatable and reliable for getting accurate positioning and for getting better quality data. Now, one of the challenges was to figure out, if you're using these two techniques together, where do you even put them on the scalp to get good coincident measurements? So we came up with a scheme where we started with the electrode positions following a clinical standard called the International 1010 positioning. So all these yellow dots represent where electrodes are positioned on the scalp. And we're looking from the top down. We've got a nose up here and ears on the side, schematically represented. So here are our electrode positions. The red dots indicate where we have deposited light onto the scalp with these optodes. And then the blue dots indicate where we are collecting remitted light from the scalp. And it's really between where the light goes in and where the light goes out that you get sensitivity to changes in the oxygen concentration. So we've made these dotted lines show the source detector pairs that overlap with the electrode positions, giving us the ability to look at correlations and how these signals relate. As a physical representation, here's a couple versions of our prototype. We also have to, of course, deal with the fact that all of our heads are different shapes and sizes, lumpy and smooth, with hair and no hair and everything you can imagine. So we have this orange material here as an elastic web that's designed to provide an isotension on the scalp. And then we have these black lattice pieces here which control the positioning. So that gives us precise positioning and repositioning of all those positions on the head. So between those two, we're able to get our coordinates more accurately and more repeatedly than other caps that just do one thing like EEG. So here we have a comparison. On the left is an assessment of the positional accuracy of a standard EEG cap, and here is the positional accuracy of our EEG NIRS cap. And what we found is that we have more precise positioning by about 40%. So we're getting more data and we're doing it in a better controlled way. We also looked at the accuracy of how controlled is the spacing between these positions on the scalp. You really want to be very careful in how you locate things on the scalp so that you can have reliable, repeatable correspondence to the brain. So here we're measuring how equidistant are these electrode locations along the scalp. And again, we ended up with a significant improvement over a standard EEG cap. So we're doing all right in terms of positioning mechanically on the scalp. So let's change gears a little bit here, and I'm going to tell you about some of the algorithm development that complements the instrumentation that we're working on. One of the key factors is to understand the sensitivity of your instrument spatially. You put these probes on the head, and can you measure everything? Do you just get the surface? Can you get all the way down to the brainstem? We need to know this in order to handle the data accurately. So we did a study. We looked at a population of 10 adult human heads, positioning the NIRS optodes on the head and simulating that light propagation down in. We calculated the depth of penetration of the light while factoring in the amount of noise we get from the instrument and from the environment and so forth. And then we considered, well, how large is a brain activation signal? So what we ultimately get is a contrast to noise. Contrast from brain function to noise from the instrument and environment. And we thresholded that here where the CNR was equal between the two. And so we know that we're able to get fairly consistently the top centimeter of the brain, the cortical layer. We can image with this near-infrared spectroscopy technique. Going down 1.5 centimeters or so, you get into higher noise, more difficult situation to accurately measure what's going on. But now we know what that is and how it varies from person to person, which is one of the pieces that you again need to have if you want to think about translation of these technologies into broader use. Another study we looked at, besides the depth of sensitivity, was the sensitivity of these different imaging methods to patterns of activity on the brain. So when you use your brain, it's not like there's one spot that's active and the rest is just quiet. There are networks of activity on the brain that vary spatially and temporally. It's a dynamic process. And every one of your imaging methods, whether it's MEG or the magnetoencephalograph, the EEG, the DOT, the NIRS DOT technique, or fMRI, each one of those is going to act as a spatial filter to patterns of activity on the brain, and not the same filter. They're all different. So if you want to study how these signals relate, you've got to understand that spatial filtering of the different imaging modalities. So we developed a contrast transfer function estimate over the brain and examined those differences between modalities. So this allows us to better understand when you see differences, it might be due to what they're measuring on the physiological level, or it could be due to how each instrument sees the brain. And so we're better able to now tease that apart. Another challenge that we faced as we dug into this is that the way we analyze the data, we usually start with a high-resolution anatomical MRI scan. So patient goes in, or subject goes in, you get a high-res MRI, and then you're going to turn that into a mathematical model of the brain to then analyze your data. Now, with something like the EEG, it's the conductivity of the tissue that really matters. That's what determines how your signal moves from down in the brain to the surface. With your near-infrared spectroscopy, it's the optical properties of the tissue. Now, you're seeing these signals through the skull and through the scalp, through the cerebral spinal fluid. You've got to have accurate modeling of how thick those layers are and how they're arranged in order to reconstruct where things happen down in the subsurface. Now, it turns out that prior work in the neuroimaging space has not really been too concerned with getting models that are robust across multiple techniques. People have been perfectly happy if they do one technique in their lab to have an anatomical model that's okay for that technique. That's not the case when you do multiple modalities. So we had to actually go back and redevelop a segmentation algorithm that takes MRI scans and breaks it up into the different components, the gray matter in the brain, the white matter in the brain, the cerebral spinal fluid, the skull, the scalp, accurately getting those thicknesses and then allowing us to apply our tissue properties, be they electrical, optical, and so forth, and get good correspondence. So these are things that you've got to do along the way. This one was another challenge. We found that, so we've got this head probe that mechanically gets our positions on the scalp, on the human subject. But then you've got your MRI, which you've built into a mathematical model for analysis. So where are those coordinates in your MRI space? One of the ways to do that is to use a positioning device, and you might have as many as 300 positions to then digitize and transform into your MRI space. We thought it would be more convenient to do the positioning in the MRI space the same way we do it with the human head, which is that we align our cap with the four corners of the head, and it mechanically finds everything else. So why should we have to do anything different digitally? So we developed an algorithm that lets us pick out the four corners of the head. It finds this top one called CZ, and then geometrically splices this mesh up and finds all the arc length subdivisions that correspond to our coordinate system on the scalp. So here in this subpanel H, we've got our 10-5, the finest divisions of our coordinate system. In blue, we've got our red showing the medium, and the yellow, the 10-20, the least fine coordinate system on the scalp. So it's coordinate systems, segmentation of the brain. There's also the need to know the correspondence between where you are on the scalp and where you are in the brain. and also knowing how that varies from person to person. So we ran a study looking at these scalp coordinates and the segmented brains, the segmented heads and brains of a population of individuals, 20 individuals. So we're mapping out, okay, here in this region, if you defined an electrode right on the top, on the crown here, this is the territory of the brain that is most proximal to that electrode. And so we map out the proximity of the electrodes to the brain in this way. We also compared that to the sensitivity, because the EEG, for example, you've got electrical currents summated over many, many neurons. Down in the brain, there's actually a directional component to that, which means there's a directionality to the measurements that you're making with the EEG on the scalp. So what is the region that is most proximal is nearby, but not identical to the region where you have the greatest sensitivity for measuring on the brain. So we're in this process trying to sort out where are we and what are we measuring with each technique. Now, for the multimodality approach, we also need to know how those sensitivities overlap between the different techniques. So here what we've done is we've taken, for every electrode location on the scalp, we've found the optimal combination of NearS source detector pairs that, when combined, give you the highest correlation with your EEG sensitivity. So the idea here is that it's a guide for saying, if you want to know how these relate, combine these NearS measurements with these EEG measurements, and you're going to get the best correlation, the best corresponding sensitivity between the two. So that's what we've done. These are basically R-square values. So in this region, sort of over the sensory motor area, we have correlations on the order of, well, R-square is on the order of 0.5 to 0.6. So it's not bad, really, in terms of the overlap in what these things are measuring. So this has been a series of techniques, algorithms applied to looking at the spatial characteristics. But again, these are dynamic signals. We are, the neuronal activity is unfolding on a millisecond time scale. The hemodynamic, those blood signals, are unfolding maybe on a fraction of a second, maybe a few hertz. You captured the cardiac rhythms, and then some. Most of the hemodynamics of the brain function are a little slower, so a few seconds, and you've sort of captured those dynamics. So we've also developed a series of algorithms to more accurately extract information from the near-infrared spectroscopy data. So in this case, I'm showing in figure A plot of the oxyhemoglobin over time for about 30 seconds of data. This is the deoxyhemoglobin changes over time. And there's a problem that can arise in this method where the absorption by the hemoglobin is a little different for the shorter wavelength and for the longer wavelength. Now that's helping us sort out the concentrations, but it's also causing the light penetration to be a little mismatched between each color of light. So that there's a factor that we use to keep track of the effective scattering of the light through the tissue called the differential path length factor, DPF. And so what we're doing here is taking the data, and in real time we can find a correction factor on the scattering of the light down in the tissue and use that to get a more accurate estimate of the oxygenation changes. Now, in a healthy subject volunteering in the lab, we're only getting a few percent correction here. But if you were to apply the same methodology to a patient who's undergone, for example, a stroke, where you could have dramatic changes in blood oxygen due to either a blockage and ischemic event in the brain, or a hemorrhagic stroke where there's a bleed in the brain, this type of technique would be critically important to having meaningful data that you can use. So if you have spatial information and temporal dynamics in your data, you can begin to ascertain how these things play out spatially and temporally. So given our neural electric data and our hemodynamic NIRS data, we're able to put together what we call a neurovascular coupling network analysis. And this is early stage results. But what I'm showing here, the circle would be, again, the same top-down view on the head. We've got a nose up top. And here are some rows where the yellow dots are EEG electrode locations, and the blue dots represent zones between source detector pairs where we're making estimates of the blood oxygen changes. And the arrows here show where we have correlations between the EEG dynamics and the blood dynamics. So it turns out these don't just play out co-localized. There's actually a spatial extent and a temporal evolution to that. So here on the left is an image showing what that network might look like in a resting brain, and just sitting quietly. And then if we were to stimulate a nerve, in this case stimulating a nerve on the wrist, on the left wrist, we get a shift in that network pattern. So this is one of the approaches to getting at the higher order dynamics in the data. So the network modeling, the other Kalman filter modeling, those were statistically based approaches. But we can also dig into the data using an applied physiological approach. Because we know something about brain physiology. It's not like it's all a mystery. In fact, we know quite a lot. And the challenge is to bridge these gaps and to utilize what we know. So here, I ran a study previously developing a model of cerebral circulation. This included blood flow being driven by the heart through the arteries and veins of the body and up and through the brain, accounting for gas exchange, accounting for what's called cerebral autoregulation, which is what prevents you from passing out when you stand up suddenly. So I've got this nice model that captures the physiology of the circulation. And there's a nice study that was published in PNAS back in 2007 by another group that dug into detail to the metabolic activity of the neurons and the astrocytes and how the metabolites are shuttled between and how those can drive the gas exchange process up in the brain. So here we're doing a modeling exercise where we're taking this finer grain information about neural metabolism and connecting it with a brain circulatory model so that we can take for example, our NIRS data and better understand how a metabolic disorder or disruption of the physiology is going to look in our data. Because we don't really know what drives these signals statistically unless we dig in either with a model like this or quite directly through partnership with another lab doing invasive readings. So this modeling exercise takes us closer to interpretation of these data. So now I'm going to take a step back towards instrumentation development. So we went through the EEG NIRS. We've gone through instrumentation, algorithms. Now we're going to look at instrumentation development in another arena where we are also doing development work. So this is enabling technology number 2, measuring blood oxygen level dependent signals with a modified magnetoencephalography system, or MEG. So we have here at Thayer an MEG machine. This unit here, actually an older system, has only 37 channels, where modern ones have 306. This is great, though, for the engineering R&D. So this system uses a very, very sensitive magnetometer called the Squid Magnetometer. It stands for Superconducting Quantum Interference Device. And all we really need to know is that it can measure with exquisite accuracy very small magnetic fields. Now, the magnetic fields coming from neural activity in your brain are so small that they get typically swamped out by the electromagnetic interference in the environment. And we've got, you might have a neural activity, even very, very large neural activity, might be only on the order of 5 or so picotesla down in the brain. And so you're going to have to go 100,000 times bigger than that to get to Earth's magnetic field. And then another 10,000 times bigger than that still to get to, say, a clinical MRI field. So we've got something on the order of a trillion-fold difference between what's happening in the clinical MRI and what's happening outside your brain due to neural activity. So huge orders of magnitude differences. So in order to measure biomagnetic fields, you need a magnetically shielded room, which is shown here in the illustration, to block out all that electromagnetic interference and allow these sensors to measure the very small magnetic fields. So unlike an MRI where you've got very high fields in a shielded room to keep the field from getting out, here we've got a shielded room to keep the noise from getting in. Now, normally MEG measures just the neuromagnetic field, but we want to do something different with it. We want to get to this neurovascular relationship. So how are we going to do it? We're going to use a material property called magnetic susceptibility. So the idea here is if you have an applied magnetic field, in this case the field line's going up, if you take a material that is paramagnetic in its property and you put that in the field, it will become magnetized along the direction of the field and create a disturbance in those field lines. If you take a material that is diamagnetic and you place it in the field, it will become anti-aligned in its magnetization with that applied field. Water, that comprises most of us, is weakly diamagnetic. Oxygenated hemoglobin is also weakly diamagnetic, almost indistinguishable from water. But deoxyhemoglobin, when the oxygen comes off, that hemoglobin becomes slightly less diamagnetic. So there's this paramagnetic shift in the deoxyhemoglobin. So we're going to exploit that to find the deoxyhemoglobin changes in the brain. Now, to find out if this was going to be possible, we started in the lab with a simpler experiment. We decided to model the brain with an inverted Erlenmeyer flask, and the change between oxy and deoxyhemoglobin with displacement of air with water. So not shown here is the MEG instrument that houses these squids up above, or the magnetically shielded room that this water pipe is passing through. So what we did is we have a simple pump here, piston cylinder pump, and I sat there. See the professor who directs the lab runs the piston pump, and my graduate student ran the instrument. So I made the water go up and down in the Erlenmeyer flask. We had these two electromagnets applied in an approximately Helmholtz configuration, creating a magnetic field across the water. And then as the water level moves up and down, we would expect to see that the magnetic field detected by those squids would be disturbed. So here in the upper plot, we've got a time axis on the horizontal, and we've got water volume in milliliters on the vertical. And over time, that water level is going up and down. Meanwhile, the applied field, starting off at 0, we're stepping it up to a little bit larger in microtesla here, up to about 30 microtesla. And just for comparison, Earth's field is about 50 microtesla around here. The squid readouts, the change in the magnetic field, when the water's going up and down, there's no applied field going across, is flat. But as the applied field gets to be stronger, the magnetic field disturbance measured by the squids gets to be larger and larger in proportion. So this was good news. It meant that we could operate our MEG in the presence of a magnetic field, and that we could measure the presence of a magnetically susceptible material. But would we be able to measure this from a human subject? That was another question. So we started with simulation to say, okay, let's take a segmented model of the human head and modulate the tissue properties of that head as if it was metabolically active and going through these changes, and figure out whether or not the signals are going to be large enough to measure. So we ran that analysis. We found that, in fact, the signal contribution from the brain is within our detectability limits on this SQUID system. At least that's what it's predicted to be. And that the signal from the brain would be, in fact, even a little bit stronger than the signal from the scalp, so long as we could involve a large tissue region, and partly because the way the brain is curved more than the scalp. So, and incidentally, the signal magnitude here, we've got 2 femtotesla, which did scare me a little bit because that's about a quadrillion times different from what you get in the clinical MRI. But we worked with that. So we ran a test with human subjects. We had 10 volunteers, and they did a task called the Valsalva maneuver, which is this. Okay, see if I... so here at time 0, they're just breathing normally, but we've got lots of people, and they're breathing sort of randomly, so their breath motions average out to nothing. They do the Valsalva, hold, release, and we measured the blood oxygen changes in the brain with our NearInfrared Spectroscopy. So we've got here a NIRS signal, deoxyhemoglobin. This is the dashed black line. And then in the black is the readouts from our SQUIDs, which nicely tracked the changes in deoxyhemoglobin. So this was our first measurement in a human subject of hemodynamics with our Meg system. Meanwhile, we had the subjects opening and closing their eyes while they're doing the breath hold thing, so that we could confirm that we can still get the neural activation changes. When your eyes are closed, you get this nice alpha rhythm in the back of the brain in particular. So we were still able to pick that up. Now, what was our resolution on that? You know, we had one, we call them a Voxel. A volume pixel is a Voxel, and that was the whole head. So not a very crisp image. So we embarked on this process to turn that susceptibility measurement into a susceptibility imager. And so we started working with a model system, which is a much more highly susceptible material, magnetic nanoparticle. Now, it turns out that imaging of magnetic nanoparticles is important in and of itself. These have applications in treatment of cancer, in diagnostic applications, in non-destructive inspection, all sorts of different areas. So here we've moved from a DC field to an AC field. We have multiple AC fields that we're feeding into a drive coil, shown schematically here. Those fields interact with the magnetic nanoparticles in the tissue, and we're measuring those field changes with in this case, a flux gate magnetometer, a much lower cost, easier to prototype system. So you would expect changes in the amplitude of the signals that are picked up and the phase of the signals that are picked up based on the magnetic properties of the particle. So because we've recognized this has value with the nanoparticle application, we're in the process of developing this into a handheld magnetic nanoparticle imager. And we're going to take all of our imaging expertise and reapply that to the brain space where we can turn our one voxel into many. So this is a photograph of our 6 voxel imager. So it's some spatial discrimination. We've got 3 drive coils here, 2 flux gates, and a couple of these compensation coils to remove noise and null out background field. And we have nanoparticles in these half milliliter Eppendorf tubes, and we can move those around in our imaging field of view. Now, it turns out that the response you get from the imaging system depends on not just the frequency of excitation, but also the type of particle you're using. So if you have a particle with a 100 nanometer hydrodynamic core, or a 40 nanometer or 10 nanometer, you're going to get a different spectral response. And so here we've mapped out when we drive these coils with AC frequencies that are, say, 400 up to 900 hertz, what is the component of the susceptibility response that's in phase and out of phase with the drive signal. So we get a whole spectrum of response. And if we take our nanoparticle samples and choose, we've got 6 positions in front of the sensor array. In this case, we had one sample at full concentration in position 3, one at half concentration in position 1. After about 20 seconds, we moved this full concentration sample to position number 4, and then we moved the half concentration to position number 2. So we're actually imaging at about 1 hertz, and we're tracking, as we're sorting these things around, not only where are the particles, when did we move them, and what's the concentration, what's the quantity of particle that's there. Now, there was some concern we set this out for review, that this would not work if we put it in a biological substrate. So we thought, what better than a maple pork sausage made in Burlington, Vermont by McKinsey. So we have our maple sausage here, and we, during the course of the experiment, we came in with a needle and we injected either nanoparticle or saline, and we can tell when were the nanoparticles injected, where were they injected, how much was injected, with a 1 hertz sampling rate. So that was a nice confirmation that it works in a biological.
So that's uncooked sausage.
Uncooked, that's right, yes. Now, our ability to separate out the spectral response of these particles also enables us to have, you could have a mixed sample, and this was just us testing this out. If you have particles of different sizes and different configurations, we can sort all that out. We can know what's where, how much, what size. And although we're only doing a few voxels, it's a proof of the concept that the math works out, the instrumentation in principle works out. So that's all ongoing. And I wanted to take just the last couple of minutes here to share with you some of the projects we have ongoing that are translational in nature, where we're taking things out of the lab and sometimes into other labs, but hopefully soon into the broader world. First one is this Neuro simulator. So this project was supported by the Department of Defense. The U.S. Army would like to use EEG to monitor, for example, alertness, attention for soldiers in the field. And this would be, say, in a Humvee driving on a bumpy road. So the electromagnetic interference, the vibration noise, everything, it's a very harsh environment. So to figure out what instrumentation and what algorithms actually work, you need a head that makes a known EEG signal. So we went about building one of those. So this is basically a plastic head that will generate a realistic EEG on its scalp. And right now we're in the later stages of a phase 2 SBIR with Creari. It's going very well, and so this is hopefully going to be on the market soon. Another project I've got going on is a collaboration with Farron Briggs and the Physiology and Neurobiology department, where we're taking, we've built a physical environment with precisely controlled, enriched visual scenes, naturalistic scenes, and a ferret is going to roam around freely in this environment with a wireless neural probe in its primary visual cortex. Meanwhile, we're going to be tracking its position with a optical position tracker, sort of like you would do with like Gollum and Lord of the Rings. So this ferret's going to be freely roaming around, exploring this natural scene. We record the visual data, and this is going to allow us to tease apart what components of the neural code in V1 are due to the scenes that go into the eye versus the behaviors that the animal engages in. So this is a way to take neuroscience, granted it's from a smaller space to a larger environment, but it's towards this direction of understanding neuroscience in a more complex world. Another project I have collaboratively taking place is with collaborators at the Psychological and Brain Sciences, Todd Heatherton and Geisel School, Jim Sargent and Diane Gilbert-Diamond, where we are doing wireless neurophysiology monitoring in children exposed to media. And the idea here is to understand how neural activity is affected by the media, as well as pupil dilation and eye tracking. So we're going to have a sense of where are they attending and what's going on in the brain. And this is going to help us to better understand how children are affected on a neurophysiological basis by media exposure. The Meg BOLD development is still ongoing. As I mentioned, we've done our first proof of concept in human. We've been developing the imaging fundamentals, and we're going to be bringing that back to the human brain. And so this is a simulation study I did figuring out what would be our depth of penetration and resolution given our current MEG instrument. This could, of course, be extended to a newer MEG to get deeper and higher resolution. But this is an active funded project. And the last study that I'm going to mention today is this one getting started this summer, where we're combining EEG and near-infrared spectroscopy to study multiple sclerosis. And this is a disease where there is they call it a clinical radiological paradox, where you have data from the MRI and fMRI studying the evolution of the plaques in the brain due to these MS lesions, as well as measuring what the brain is doing during different tasks. And there's a disconnect between the clinical symptoms and what can be measured radiologically. And it's not really understood why. Now, we believe that the explanation is going to come from understanding how the blood dynamics measured with fMRI are related to the neural dynamics. And we're going to be able to tease that apart by combining EEG NIRS in these MS patients. So we're going to look at MS patients and healthy controls and try to understand how this neurovascular coupling might get altered in the disease. So the multimodal neuroimaging laboratory, how are we bridging the gaps? So where a single modality approach would study a single aspect of brain physiology, combined EEG NIRS or combined Meg BOLD help relate the neural activity in the hemodynamics. Whereas a single modality approach would operate at a single spatial temporal scale, in particular the magnetic susceptibility approach has the ability, the potential at least, to bridge between multiple scales spatially and temporally. Now I haven't gotten too far down this particular path, but I know fundamentally that the physics of the measurements we're making can be scaled down, can be scaled up, and it should work equally well as a function of scale. So that enables it fundamentally to bridge scale. And whereas these techniques measure detailed anatomy, measure detailed physiology, it's really through the translational research projects that this neuroimaging research connects the data to human needs. And that's where ultimately this work has impact. So with that, I would like to acknowledge and thank the many contributors, the members of the Multimodal Neuroimaging Laboratory. We have many women and men who are contributing to all of these projects in the lab and keeping it lively, keeping it fun, and keeping the research moving forward. I'd also like to acknowledge the many funding sources that have supported the lab over the years here at Thayer. There's been a really broad mix of funding sources, internal and external, that have supported this work. And here's another aspect of the lab from the 2003 Thayer Fitness Challenge. So with that, I'd like to open up to questions. Yes, Bob.
Yeah, I have a fairly basic question about power consumption because it seems that it actually be too difficult to measure. But the more I thought about it, first of all, there's a blood-brain barrier, is there not? So the blood vessels don't actually go into the brain. Does that imply some storage areas in the brain for energy? And then there be consequences of time delays between activity and power consumption because you couldn't assess what was in storage.
Right. Right. So the way that the neuron deals with this power budget is it's like a preloaded spring. You've got all the energy necessary for that neuron to fire an action potential is stored in the chemical potential difference between its inside and its outside.
So the whole brain is sort of loaded.
The whole brain is loaded all the time. And then when a neuron fires, gets too excited, sends out an action potential, then you've got this active process of rebuilding that state of readiness. Now the energy, the oxygen that's supplied to those vessels can go right through the blood-brain barrier. The oxygen, CO2 freely diffuse through. It would be the larger molecules that are being blocked by the blood-brain barrier. So there is a fairly tight linking between oxygen consumption and the metabolic activity in the brain. Though as you examine it more closely, it does get more complicated. You get other involvement of lactate and shuttling of lactate between the astrocytes and the neurons, and so things do get metabolically more complicated. And we do have tools to study metabolic effects. For example, PET, the positron emission tomography, with an injection of a radioactive oxygen, can be used to track that oxygen consumption. So it's an area of deep study. It's one of the critically important aspects of understanding brain physiology and how diseases affect the brain. And it's an ongoing area of study for both the measurement and interpretation.
CSF would then act as a source within the brain that's saturated with oxygen already?
Well, it's really at more of a distance. So the diffusion length to supply oxygen to the cells needs to be fairly small, because it's a slow process at long distances. So that's really coming from the capillaries down in the beds. So that CSF is more of it's cushioning and protecting the brain, and it is just helping to keep things clean and clear there. Yeah, Alex.
I didn't understand from your presentation, and I may have missed the detail. When you do the analysis of your sensitivity region or the zone of influence, do you actually use a patient-specific model in each case, or do you have a sort of a template that will tell you that?
So we use a patient-specific model in every case, and what we'll do is we'll average those results over on the order of 20 individuals. And there are atlases of the brain, but any atlas that has detailed anatomical structure is going to be derived from a single head. Maybe it'll end up being warped to make more of an average geometry, but there is considerable anatomical variation in the morphology of the brain between individuals, not just bulk size, but the whole way that it's folded. There are certain, we call the creases in the brain, the sulci, and the bulges, the gyri. And there are certain large ones, like one called the central sulcus, that's generally easy to find even for someone who's not a neuroanatomical expert. But the details of how the brain is shaped and folded and configured, highly variable between people. So if you have an average brain, that's not going to be representative of an average person in a way. Other questions? Elsa, yes?
Is there any way to represent a scale? Is there any sort of minimum scale that you really want to reach to be useful, or ultimately you have to go all the way down to per neuron?
So it depends on what you want to know. So the question is there an average? Is there a scale you need to be at? And to understand brain function on a coarse level of what territories are involved, being on the centimeter or so scale is okay, and that can even allow you to look at networks that extend over larger brain regions, which many do. A motor system, for example, is going to involve primary motor and supplementary and premotor areas that can be centimeters apart from one another. But the thing is, there's more and more information. As you get down to a finer scale, you reveal more information. And all the way down to the cellular level and to the synapse level, you've got more information about what the brain is doing. So now, to a certain extent, this is like building a map. If you want a map of the U.S., you don't want to have it the same size as the U.S. because it's not particularly helpful. Last summer I folded it. It's like so. But that doesn't mean that you don't want to be able to zoom in to get an answer to a very specific question. In a disease process like in Alzheimer's disease, you're going to have plaques and tangles down in there on the cellular level, mucking up the neural function, mucking up the vascular function, and that's it's going to be important at the fine grain and the coarse grain. Charlie.
Of all these, following up on that, of all these different methods, what method has the best hope of getting to the finest resolution, and what limits that?
So, right. So the question is, what method to get to the finest resolution? So it depends on what level of invasiveness you're willing to tolerate. So, we already have highly invasive methods of getting extremely fine-grained information, but it's not always appropriate for use on a healthy volunteer, right? So what's the, there's a scale. It depends on who is participating in either in the research or using the technology. If, let's say an individual didn't have use of his or her legs, and maybe then the bar would be different for a neural implant that would have fine-grained enough information to fully control robotic legs. Now, an individual who's looking for, looking to get away from their mouse and just navigate their computer might not want their skull opened up. But you might tolerate, say, an EEG cap. Now, we have right now a lot of advancements in wireless EEG, where you could go out and buy for a few $100 an EEG system that would measure a few channels. Whether it gives you the resolution you need to be productive on your computer is still a little hazy, but we're getting closer. So really, we'd like to be a little bit finer on the resolution, better sensitivity, lower noise than anything available now. I believe that we're going to get there with the combination of the magnetics imaging at multiple scale. I think that's going to really move us forward. Yes.
Fascinating examination of our imaging process. How do you see this discipline beginning to interface with more therapeutic efforts like transcranial magnetic therapy?
Right. So it's a great question, because this is the imaging side, and there's the whole intervention side as well. And they do overlap on many levels. And one is just guidance of any kind of intervention. There is the same mathematical modeling that I use to figure out where can you measure with the EEG. Those are the same models you use to figure out, well, if you put electricity into the scalp, where is it going to excite the brain? And the same is true with the magnetic side. So these equations go both ways. The hardware would have to be adjusted, for stimulation versus readout, but they're very much related. Another aspect of it is, say someone has gone in and received a deep brain stimulator to deal with their Parkinson's, for example, or possibly in the future even for intractable depression. They might have a stimulator implanted in the brain, or epilepsy, let's say. Those are not typically fMRI compatible. So what if you need to get imaging data from the brain from this person who has a large chunk of metal inside their head? So there's a need there for imaging methods that are tolerant and not going to cause any injury, which would include, for example, the optical methods, the low-field magnetic methods, and so forth. Other questions?
One more question. Why did you pick on Eric?
Oh.
These may be way ahead of you, but do you and or your colleagues understand how the brain stores information? And if not, will any of these methods eventually show it?
Okay, so the question is, do I understand how the brain stores information? Not very well. But there are experts here at Dartmouth who specifically address the question of learning and memory in the brain. And these imaging tools are really essential to advancing that science. So I have collaborators and soon-to-be collaborators as these technologies mature here at Dartmouth in psychological and brain sciences, for example, to look at learning and memory. I'm also interested, and I've got one of my...
These methods that you're describing will eventually get down to that?
Well, it's going to be a combination approach. I mean, you're going to... Learning and memory is another one of those brain processes that bridges across scale. I mean, on the large scale, there is network level involvement of multiple brain centers for receiving, processing sensory information, turning that into a context, deciding somehow through this neural processing that a piece of information needs to be retained, and then as that gets, say, stored more long-term in the hippocampus, the long-term memory storage region in the brain, how that gets instantiated into the neurons is through a neural plasticity process. So there's actually a rewiring, a reweighting of the synaptic connections, a rearrangement of how those neurons fire together that is at the cellular level and how individual neurons interact with one another. So to get the whole picture, again, you're going to need to bridge scale. You're going to need to understand how these different physiological elements relate, and then ultimately relate that to the human experience of memory.